Hi, everyone. Good morning. Thank you for that introduction. First and foremost, I would like to thank PEGS for giving me the opportunity to present here on behalf of Eurofins DiscoverX. Again, my name is Venkatesh Chari, and I'm the Scientific Market Development Manager at Eurofins DiscoverX. So today I would like to talk to you about our assays and how Eurofins DiscoverX is helping accelerate immuno-oncology drug discovery with MOA-Reflective functional cell-based assays. So I've divided my talk into 4 parts. Firstly, I would like to give you a brief introduction about our company, who we are and what we do. Next, I would like to give you an overview about the core technology that we use to build all our cell-based assays. So speaking here at PEGS, we do understand that we are talking to a very broad audience, and hence I would like to give you an overview about some of the really exciting stuff that we've been doing in the last 8 to 10 years. And this would be focused around the large molecule assay portfolio that we have. So I'll be talking about some of these assays that we have for checkpoint receptors, bispecific antibodies, cytokines, interleukins, and the cytotoxicity assay platform that we have. And to conclude with, I would like to walk you through about our strategy on how we provide end-to-end support for QC lot release testing programs. So this is us, Eurofins DiscoverX, and we are headquartered in Fremont, California. So DiscoverX was founded about 20 years ago, just a little before the year 2000. And recently in 2016, 2017, we were acquired by Eurofins, and now we are a Eurofins group of companies. So for the past 20 plus years, we've been developing functional MOA-reflective cell-based assays. And hence we have since we've been doing this for such a long time, we have the industry's largest cell-based assay portfolio in over 10 plus druggable target classes. So building assays is one thing, but providing support is another. Hence, we have a global FAS team that would work with you to provide end-to-end support to ensure customer success. So when we develop an assay, we have this FAS team that would work with you or the receiving site to have the assay transferred to that site. Apart from that, we work with global CROs. We have a CRO certification program in place. And by that, what I mean is that we've been to their site, we've performed virtual trainings, and we've ensured that they have the expertise and the right kind of equipment to run our assays successfully. Our assays are ICH-based qualified, and this makes them fit for purpose for being implemented into GMP QC lot release testing programs. And lastly, we are supporting 100 plus global programs for potency, stability, and NAB testing. And this way, Eurofins DiscoverX is enabling biopharma to bring better drugs into the market faster. So recently, in 2021, FDA approved the 100th monoclonal antibody drug product. And we are in the middle of 2023, and we are already inching towards the 200th antibody product to be approved. So that is the pace at which the biologics market is growing. So we are very familiar with a lot of the molecules that have different kinds of MOAs or mechanisms of actions. And here we have a few examples. So you have an anti-receptor like Keytruda, which is an anti-PD-1. You have anti-ligand antibodies like Avastin or bevacizumab. Then you have agonistic antibodies, bispecific antibodies, and so on. And all of these rely on disrupting ligand-receptor interaction, excepting for agonistic antibodies, which pretty much activate the receptor. And now on the other side, things are a little more complex, where you have effector-mediated function. So, for example, you have rituximab, which is an anti-CD20 antibody, or you have Blincyto or blinatumomab, which is a bispecific T-cell engager. And both these kinds of molecules, they essentially bring in the effector cells that engage with the target cells, which causes target cell killing. So our assays are flexible and adaptable to assess all these different types of mechanisms of actions that you see here with these different types of molecules. So, so this is the way that Eurofins DiscoverX is supporting the large molecule drug discovery programs, and that is going to be the focus of my talk here today. So the core technology that our assays use, it's a very simple technology. It's enzyme fragment complementation. So it's based on a split beta-gal enzyme system. So what we've done is we've taken the beta-gal enzyme and we split it into two parts. One part you see here in orange and the other one in blue. And individually, these fragments are inactive. However, when they come together, they form an active enzyme complex. And when you add substrate, the enzyme hydrolyzes the substrate and gives you a nice light signal, which is a chemiluminescent signal. So how do we use this in our cell-based assays? So imagine you're developing an assay for PD-1. You have the PD-1 receptor on the cell surface, and then you have the SHP2 protein in the cytosol. So we've tagged the PD-1 receptor with one of the EFC tags and SHP2 with the other. Now, when you add ligand, or in this case, PD-L1 ligand-presenting cells, this activates the PD-1 receptor. And what this does is it recruits SHP2 to PD-1. So SHP2 is known to phosphorylate PD-1 as an activation mechanism. And this brings the EFC subunits together, and it forms an active enzyme complex. And when you add substrate, you see a nice light signal, and you get a nice gain of signal kind of an assay. Now, in the same assay, when you add a molecule like Keytruda, which is an anti-PD-1 antibody, you get a complete inhibitory curve within the same assay. So it's a very simple technology, the EFC technology, but we've built very sophisticated assays to address such complex biologies. Our assays have a lot of different advantages as well. They're homogeneous. So it's a simple add and read protocol. There is no wash steps, or you don't need to spin the plate. The assay is extremely sensitive. It's enzymatically amplified. And it's a pretty robust assay, generating a really large dynamic range or assay window, or signal to background ratio. And one of the things that is advantageous is also it being a chemiluminescent readout. You really don't need any sophisticated equipment. A simple benchtop luminometer will do. Last but not the least, this is very important that our assays are extremely easy to transfer between sites. So our assays having all these different attributes make them really easy. So when we develop an assay, the idea is that the assay should work with the same precision and accuracy in your hands the way we've developed it with. And all these different attributes make it really easy to transfer from one site to another. So again, using this simple EFC technology, we've developed a lot of these different types of assay platforms. So you have protein-protein interaction assays that we, for example, the one that we just went over. We have receptor dimerizations, we have downstream signaling reporter assays, cytotoxicity assays, and so on. So this is a nice snapshot or glimpse of our assay portfolio. And we have assays for GPCRs. And we just saw in the previous talk by Chris where he presented a data set using our GPCR assay. So pretty much Eurofins DiscoverX started off as a GPCR company back in 2000 when GPCRs were really hot and everyone was working on small molecules for GPCRs. So pretty much we built assays for all the GPCRs under the roof. We have assays for 289 unique GPCR targets. And when we developed these assays, we developed assays to assess multiple different mechanisms of action for each GPCR target. So for each GPCR target, we have cyclic AMP signaling assays, GPCR beta-arrestin recruitment assays, GPCR internalization assays, and also calcium signaling assays. So what this helps is it helps researchers cross-validate their results. So pretty much there are only so many GPCR targets out there, and in the first 3 to 5 years, we built assays for every GPCR out there. But we did not stop there. We went on to build more complex assays for checkpoint receptors, cytotoxicity assays, signaling reporter assays, and then we have assays for cytokines and interleukins. And during the pandemic, DiscoverX really focused on developing assays for every cytokine that was implicated in the cytokine storm as a result of COVID-19. So all these assays that we have, they are all functional and MOA-reflective, and that makes them really well acceptable by regulatory bodies. So, and here is a slide to show you from a variety of different reports, like the FDA, EMA, and others, that our assays are a part of IND filings, BLA filings, patents, and so on. So there are a number of reports and publications that show our assays are acceptable as MOA reflective to go into GMP lot release testing for eventual commercial drug release of the drug product. So now I would like to go over some of the key large molecule assay portfolios, starting with the immune checkpoint receptor assays that we have. So this is a glimpse of the assays that we have in our checkpoint receptor portfolio. So it's quite an elaborate portfolio, and we have multiple different kinds of MOAs that we cover. And this includes the co-culture assays or co-culture models. For example, we have assays for SERP-alpha CD47, PD1, PDL1, like we just went over, BTLA, HPM, and others. So the idea here is to capture the signaling axis, and hence we developed these co-culture models so that it's extremely functional or physiologically relevant and MOA reflective. One of the other things that we worked on developing are these clustering cell lines, and what these do is they help or aid in increasing the activity or enhancing the signal that you see with agonistic antibodies, for example, against checkpoint receptors. And apart from that, we have assays for the TNFR superfamily, pathway signaling assays, and I'll be going over a few of these in the next slide. So here, I wanted to show you a variety of different MOAs that you can assess using our checkpoint assays. For example, these are assays for anti-receptor, anti-ligand, agonistic antibodies, plate-bound ligands, clustering and cross-linking. And also, apart from biologics, our assays can also be used to characterize or rank order potency small molecules as well, like you can see in the bottom right panel over here. So what I would like you to focus here is you can use the same assay to assess multiple different kinds of MOAs for your molecule. For example, if you look at the top two panels here, it's the same PD1 signaling assay. And the data on the left shows you how we've used this assay to characterize an anti-PD1, which is an anti-receptor antibody. And on the right, you can see the same assay being used to characterize an anti-ligand antibody, which is an anti-PDL1. So essentially, it's the same assay assessing a couple of different MOAs. So now I would like to talk about the assays that we have for characterizing bispecific antibodies. And this has been an area of focus for us in the last 5 years. So again, it's a pretty simple assay system. For example, you have one arm of your antibody binding to PD1, the other arm binding to CTLA4. So what we've done is we've taken these cells and we've tagged the PD1 receptor with one EFC tag and CTLA4 with the other EFC tag. And if your bispecific antibody binds to these targets, it will essentially bridge these two receptors, bringing these two EFC tags together. And this forms the active enzyme. And when you add substrate, you get a nice gain of signal assay. And of course, if your antibody or molecule is non-specific, you're not going to see a response. It's pretty much a flat line that you would expect. So now imagine you're screening hundreds of different kinds of molecules as a part of your screening program. You can take a simple assay like this and run either in a 96-well or a 384-well assay format. And you can nicely rank order potencies for the molecules that you're screening. So the point that I'm trying to make here is that the assays are extremely scalable and flexible to assess all these different kinds of things. And similar to PD1 and CTLA4, we have assays for different kinds of bispecific molecules like the PD1 LAG3, PD-1 CTLA4, PD-1 PDL1, and so on and so forth. So here is a nice customer data set that we created while screening bispecific molecules. But what you can see here is that only the bispecific antibodies here are able to generate a nice dose response curve. Whereas the monospecific antibodies or MSABs, individually or in combination, they are not able to produce any response, and you see a flat line over here. So only the bispecific antibodies are able to bring these receptors together, which results in the formation of an active enzyme or EFC enzyme complex that gives you a nice signal. So next on my list, we have assays for cytokines, chemokines, and interleukins. And this is a nice bird's eye view of our cytokine assay portfolio. Now, a lot of them you see are interleukins. And we have assays covering every target class of interleukins out there. And here you can see that the interleukins have been pretty much grouped into 7 to 8 different categories. So we have assays for popular targets like IL-2, IL-10, IL-12, 23, 17, and so on. And then we have assays for other cytokines as well, which includes another set of popular targets like TNF-alpha, IFN-gamma, GM-CSF, GCSF, and so on. So apart from interleukins and cytokines, we have an assay for a whole bunch of chemokines, which are classically GPCRs. And since they are GPCRs, we have assays that address different MOAs. So for every GPCR target, we have a cyclic AMP signaling assay, beta-arrestin recruitment assay, and a GPCR internalization assay. So this is how the assay looks. And what I would like to show you here is that you have a robust response if you're running the assay in an agonistic mode or an antagonistic mode. For example, we have assays for ustekinumab, tocilizumab, and anakinra. And all of these produce a nice agonistic curve. With IL-23, for example, in the ustekinumab assay, you see a nice agonistic curve with IL-23. And with the antibody, you see a complete inhibition in the same assay. And you see similar trends with the other two assays that we have as well, that they are able to generate an agonistic assay, and in the same assay, you see a complete inhibitory curve with the antibody. So many of us are familiar with signaling reporter assays, and we've been using this for many, many years. We are very familiar with STAT3, STAT5, NF-kappa-B, and NFAT reporter assays, and we have those too. So the way this assay works is the ligand binds to the receptor, kicking off a signaling cascade, which causes the transcription factor to enter the nucleus and bind to its regulatory element, response element, and this causes downstream gene activation. And what we've done in these cells is this particular downstream gene, we've taken a housekeeping gene and tagged it with one of the EFC tags. So once that gets expressed, you add the detection reagent, which has the lysis buffer, the substrate, and the other EFC tag. And this brings the two EFC tags together, generating an active enzyme, giving you a nice chemiluminescent signal. So here are 3 different assays, or assays for 3 different kinds of reporters that we have. We have NFAT, NF-kappa-B, and STAT3. And these are some of the most common ones that you see on the field. So we've developed nice, robust assays for these. For example, you see a nice agonistic curve with the anti-CD3 antibody in the NFAT reporter assay. And similarly, you see the same trend in the other 2 assays as well. And now I'm showing you how these assays were used to characterize 3 of the FDA-approved drugs, such as tocilizumab or Actamra, Humira, and Prolia or denosumab. Again, even here, you see a nice complete dose-dependent inhibition with these molecules. So these reporter assays that we have developed, they serve as complementary assays to the dimerization assays that we have for interleukins. And last but not the least, I would like to talk about our killer assay platform. Now, this is our main portfolio to look at cytotoxicity. So across all these assay platforms that we have, this has got a lot of attention recently, as it can assess multiple different things in regards to cell-mediated cytotoxicity. So basically, this platform is very versatile, and it covers many different applications like ADCC, ADCP, CDC, T-cell redirection, which is also referred to as T-cell engagement, or TDCC, which is T-cell dependent cytotoxicity, and CAR T-cell therapy as well. So using one single assay platform, you can assess multiple different applications for your molecule. So for example, rituximab, which is an anti-CD20 antibody, has an MOA through ADCC, ADCP, and CDC. So you don't need any secondary assay. You can use the same assay to assess multiple different applications or modalities of your therapeutic molecule. So we have similar data sets for all these different applications. However, due to time constraints, I would be only focusing on the ADCC assay that we have. And how does this assay work? So again, it's pretty simple. So imagine you have a target cell with a particular receptor of interest, let's say CD20. That's one of the popular targets. And then you have rituximab, which is an anti-CD20 antibody. And let's say the target cells are Raji cells that express CD20 endogenously. So what we've done is we've taken these target Raji cells and we've modified them to express, stably express a housekeeping protein, which is tagged with one of the EFC tags. We refer to this protein as the killer reporter protein. So we've generated 40 plus cell backgrounds, which already express this killer reporter protein. And if there is any other particular cell background that you're interested that may not be on our that may not be available off the shelf from us, what we do offer is also retroparticles that you can take in-house and build a stable cell background of your own choice. So next, you would add your target-specific antibody and your effector cells. And your effector cells could be any one of these listed here. And as you can see, it's a very open platform. You can choose a cell background of your choice or use effector cells of your choice. And after the antibody gets opsonized onto the target cell, the effector cells engage with the FC part of the antibody, and this results in ADCC activity, which kills the target cells. So once the target cells are killed, the killer reporter protein gets released into the assay media, and then you would add your detection reagent, which would have the substrate and the other EFC tag, which would result in an active enzyme complex, giving you a nice light signal, thereby giving you a gain of signal kind of a response. So you see a nice dose-dependent increase in activity, which correlates with increased ADCC activity or percent killing of the target cells. One of the things to note here, sorry. So next, what I would like to show you here is example data sets that we have. So the first panel over here shows ADCC activity for CD20 using rituximab in all these different cell backgrounds. We have ARH77, Dowdy, Ramos, and Will2S. And you see that it generates a nice kill curve in all these different cell backgrounds. So if you look closely, the curve shape for all these different cell types is slightly different. So what we did is we quantified the amount of CD20 that was expressed in these different cell types. And the ADCC activity really correlates well with the CD20 density on these different cell backgrounds. So similar to CD20, we have assays for CD38, CD33, HER2, and EGFR with cetuximab, and so on. So one thing to note that in these assays, the target receptor of interest has not been overexpressed. So all the ADCC activity that you're seeing here is a result of the endogenous receptor expression. So to keep things very physiologically relevant, we haven't overexpressed the target receptor. One recent development that we have is our focus on T-cell engagement, which is again also referred to as TDCC or T-cell dependent cytotoxicity. So you have your BiTE molecule or a bispecific T-cell engager. One arm engages with the target cell and the other arm engages with the effector cell. This results in target cell killing mediated by the effector cells, releasing the killer reporter protein into the assay medium. So very similar to how it works for the ADCC assay of ours. So this is a nice data set that we generated using blinatumomab or Blincyto, which is a BiTE molecule. And this was done in a Raji cell background. And you can see that it generates a really robust kill curve, giving you a really nice high assay window. And for this assay, we used the really well-established pan T cells as the effector cells. Now, this is another data set that we generated using the same cell background. And here what we did is we miniaturized this assay into a 384-well assay format, and we screened for potential T cell and candidate T cell engagers. And we were able to generate a nice rank order potencies for all these different samples. So essentially, this assay is extremely scalable, and you can also run this in a 384-well format to either characterize your molecule or to perform any kind of screening or rank ordering your candidate samples or molecules. So here in the next section, I would like to show you our strategy for providing end-to-end support for QC lot release testing and commercial release of the drug product. So the first step begins with generating the cell line. Now, this is the most time-consuming step, but we have, since we've been building cell-based assays for so many years, there is a good chance that the particular target of interest that you're looking, we already have an MOA-reflective cell line available in our portfolio for you. So that would pretty much save you about 5 to 6 months right there. So the next step is to take this cell line and then convert it into a ready-to-use bioassay format. And we have a very established process for this, and we are very experienced in converting cell lines into the ready-to-use assay formats. We've done this for hundreds of assays. I believe we've done this for about 600 to 700 different assays in our portfolio. And if the molecule that you're working with, if it's a biosimilar, then there's a good chance that we already have a qualified bioassay available off the shelf for you. And this would save you additional time in regards to the whole process. Now, the next step is very important. What we do is we would take your drug molecule and optimize this assay for that molecule. Following that, we would perform assay qualification based on ICH guidelines. And as a part of the assay qualification, we'll be looking at all these good things like accuracy, precision, range, deletion, linearity, and so on. And we've already qualified assays using FDA-approved drugs like Humira, Actemra, Keytruda. Only a few of them are listed here, but we do have more. So all these, the first 3 steps would be performed at our headquarters in Fremont, California. So the next step is to transfer this assay. Now what we would do is we would take this assay that we've developed and transfer it to the receiving site. And this receiving site can either be your site or an affiliated CRO of yours. And I'm personally involved in this step. So me and my team have a very dedicated process to take this assay and to transfer this method to the receiving site. After that, the CRO would take this assay in, implement it into GMP, perform validation, and subsequently perform routine lot release testing for the commercial drug release of the product. So as for providing long-term support, we do critical reagents management. We have well-characterized analytical cell banks. We do supply agreements, and at any given point, we are fully audit ready. So this is my last slide, and here what I want to show you is that we have a number of assays belonging to different target classes that are a part of routine GMP testing that includes potency, stability, and NAB testing for commercial release of drug products in different regions. And this includes North America, Europe, and the Asia-Pacific regions. So again, like I mentioned earlier, since our assays are extremely MOA-reflective and functional, they're very well accepted by the regulatory bodies for GMP lot release testings. And this is the proof of that. So in conclusion, we at Eurofins DiscoverX have the largest portfolio for cell-based assays, and this includes off-the-shelf products to support immuno-oncology therapeutic development. We have path hunter cytokine and interleukin assays that cover over 85% of the targets. And then we have our killer cytotoxicity assay platform, which you can use to assess multiple different applications like ADCC, ADCP, CDC, T-cell redirection, and CAR T-cell therapy. And lastly, we have a very good strategy for providing end-to-end support for biologics drug discovery programs. And this is how we are enabling biopharma customers to bring better drugs into the market faster. So if you have any specific questions or want to take your discussion further, we do have an exhibition booth here. Our booth number is 511, and we hope to see you there to take this discussion further. With that, I'll stop here and happy to answer any questions you may have.